Dr. Aaron Sato is a well-known friend of ours who shares the same emphasis and passion for science storytelling that we do here at BioRender. With an extremely impressive career in a number of biopharma companies, Aaron is the author of over 30 peer-reviewed papers and has 50 issued patents in the antibody space. He is now the CSO of Twist Bioscience, leading the Twist Biopharma division, which provides end-to-end antibody discovery and optimization solutions utilizing its proprietary DNA technology to write synthetic libraries. We are grateful to have him join us today to share his expertise from Twist Biopharma and how BioRender helps his team communicate their work. Without further ado, Dr. Sato.
Thanks, Jeff. Can everyone hear me okay? All right. So again, my name is Aaron Sato. I'm the CSO of Twist. As Jeff said, I'm really happy to be here today. I'm going to give an overview of all the great things we're doing here at Twist, and of course, use a lot of pictures to convey my messaging, since I'm a big advocate that really a picture tells 1,000 words. So we'll go through the story of Twist and then work through and kind of go through the journey that I've also gone through. I really appreciated Don's presentation before this, where he actually started off with his own story. I always say that the one story you should always be able to tell is your own personal story. And so I'll also do that as well in my presentation today. So next slide. So I'll start off with that. So again, in terms of my career path, again, you can move through the various. I'll start off with where I grew up. I actually grew up in Kennewick, Washington, which is in southeastern Washington State. I went to Kamiakin High School, again, where the number one subject was probably football. But surprisingly, I was able to find a lot of great teachers there. It really inspired me to get into STEM subjects and really learn to love math and science. And so from there, I went to the University of Puget Sound, where I actually majored in chemistry. Again, a small liberal arts school. I'm really a big advocate of small liberal arts education because it really helped me to understand how to think well. And from that, I really fell in love with chemistry, and so decided then, next image, to go to the Massachusetts Institute of Technology for my graduate degree, where I got my PhD in chemistry, studied structural biology, worked in a lab focused on discovering X-ray crystal structures against immunological receptors. And so again, really spurred on my passion for understanding structure, function, relationships. Next image. I then went on and worked for Dyax Corporation. Rather than do a postdoc, I went directly and did work for a biotech company, also in the Cambridge area, very close to MIT. This is actually my ID that I love to show. I often try to comment that I don't look that dissimilar from my photo from over 20 years ago. But again, this is my very first job, and I learned at Dyax how to basically apply science in a biotech environment to actually help discover and create biologics. So it was a really great learning experience for me. Next opportunity I had is I went and then worked for Oncomed, which is a cancer stem cell company in California, where I actually, again, served as a service function within the company to help them discover biologics. This is a picture of me giving Jerry Hill a tour around the lab. This is actually an image of a pamphlet that keeps showing up every year when Jerry Hill goes up for re-election in the Bay Area. So I keep seeing myself on the cover of this pamphlet that he loves to show. As you can see, I'm super excited to be giving Jerry Hill a tour around the lab. Next image. I then went on for my first executive role to actually work at Sutro Biopharma, where I, again, spurred on my passion for antibody drug discovery, learned how to do a lot of antibody drug conjugate work. And so again, really loved that opportunity as well in the Bay Area. Next slide. But today, I actually work at Twist Bioscience, which is an amazing DNA company. I'll get into more around the base technology for it, but this kind of shows you some images of my journey through Twist. We IPO'd over 3 years ago. I now have an amazing team that focuses on using all of Twist's amazing DNA technology for antibody discovery and antibody optimization. Next slide. I always ask people, what was the singular moment where you kind of figured out what you wanted to do? And for me, I mean, that journey I talked about before was important, and all the teachers are really important. But for me, I can always remember back to that one moment where my grandfather actually gave me a chemistry set. And that moment was the genesis of me like falling in love with chemistry. And so I always love to show this. This isn't the actual exact chemistry set he gave me, but it just really shows you that, thinking back to that genesis moment where you kind of knew what you wanted to do. So I love to show this image to really help convey that. And that's something that I think everybody should really stem and try to find their moment where they really understood what they wanted to do. Next slide. As kind of an aside, I love to collect chemistry sets now. I actually have multiple chemistry sets. I love to find kind of like antique chemistry sets. This is actually the very first chemistry set that was actually ever created in the United States. It was made by an American chemist back in 1914, and it was called Chemcraft. I love to point to some of the kind of notes on some of the advertising. One of them is that it contains no phosphorus or hazardous substances in the kit, which I probably wasn't true. There probably are a lot of hazardous chemicals in this chemistry set. And I've actually always been on the lookout for a Chemcraft chemistry set. So if anybody ever sees one, definitely let me know, and I'd love to get my hands on one of these. But I do have some kind of later generations in my office in Burlingame. Next slide. And then another thing I love to point to are kind of like the passions in your life. And so for me, it really is kind of three primary passions that really drive me. One is, of course, my family. Really big on doing family events. This is actually a picture of my daughter and my son where we actually went out and did a llama hike. I really love llamas because they have these really unusual antibodies that I'll talk about later. But so this really combined my second passion, which is, of course, antibody discovery and working in biotech. This down in the bottom right-hand corner is a picture of me with my team at Oncomed, where we actually discovered a bispecific antibody that was actually still in the clinic today that targets both VEGF and DLL4. And then finally, my last passion is, of course, skiing. I'm a huge skiing fan. I love to ski, and I love to also combine that with my work, hanging out with my family. So it's and I love to travel all around the world and ski different locations. Next slide. This is just going to really simplified view on how do you make a protein. I like to show it when I give kind of presentations to other non-scientific audiences, but I think, again, it just brings home the message that everything comes from DNA. So all proteins in your body, all organs, everything starts with DNA. And so it really shows you that working in a DNA company like I do really shows how I have this amazing tool at my disposal to basically make amazing proteins like antibodies. Next slide. And so how do we do that? Okay, we use oligonucleotide synthesis to make oligos. And again, using conventional oligonucleotide chemistry, we can make oligos up to, say, 300 base pairs in length. And again, the way you make a gene is basically make a bunch of different oligos that span that gene, let's say a 1.5 kb gene upwards to even 5 kb genes. And we basically make all the oligos that constitute that gene, and then we can basically splice them all together to make a gene. And as you'll see in a second, we can then use the Twist platform to make oligos really quickly and in large numbers to enable folks to make lots and lots of genes. Next slide. I'll also talk about antibodies as it relates to DNA as well. So again, antibodies, as you may know, are in your immune system. They're really, again, amazing proteins that basically serve a lot of different functions. I think you probably have heard a lot about them over the last couple of years with the COVID-19 pandemic. And again, the vaccines that we all have gotten basically promote the formation of these antibodies so we can clear out a lot of pathogens like COVID-19. But they also serve all kinds of other functions to basically address specific diseases, attack specific cells you might want to get rid of. And the idea around Twist Biopharma is we have this amazing ability to create synthetic libraries where we can actually discover directed, targeted antibodies that do a lot of the same functions that antibodies normally do in our own immune system. Next slide. I always love to cite kind of the main advantages and maybe potentially disadvantages of antibodies as it relates to, say, comparing to small molecules, which are probably more common therapeutics you probably think about. Again, advantages of antibodies is that they are, again, very interesting proteins. They have multiple domains. So they have a Fab domain, which is the main region that binds the specific target that they bind to. And then they also have an FC region, which is kind of at the bottom of the Y within an antibody. And that actually, those two regions of an antibody actually convey many of the advantages that are shown on the left, in the sense that they are highly specific. They bind very specific targets. They don't bind a wide range of different targets. They're often very focused on one particular epitope on another protein target. They have a very long half-life. So unlike a small molecule that you might have to take every day, an antibody you might actually only have to take maybe once every 2 weeks or maybe once a month. There are even some therapeutics you only have to take like once every 3 months. And that's because the FC domain of an antibody actually conveys, it has a long half-life and actually conveys, allows it to stay in the blood system for a long time. And then antibodies can also be used for all kinds of specialty applications like delivering drugs, delivering highly potent toxins like ADCs. And you can also even make like really complex antibodies that might bind multiple targets and create bispecifics. Some of the disadvantages of antibodies is they're sometimes very costly to make, unlike a small molecule. Some small molecules typically are a little bit cheaper to make than an antibody. You sometimes can find that there's some toxicity associated with antibodies because sometimes they're very specific. And because of that specificity, they can actually are sometimes very potent and directed against specific targets. And that can sometimes lead to cascades of responses in your body that can lead to some toxicity. And then finally, because they're proteins, it oftentimes limits the route of administration, unlike a pill, which you can oftentimes swallow. Next slide. So just again, looking ahead to the future, Twist Biopharma, we're a division of Twist Bioscience. I'll get into the second, the kind of the details about what we do, but we're really focused on helping others discover novel biologics. And we're looking for folks to help them, give them an advantage to help them discover and optimize drugs very quickly so that they can go out and help patients. Next slide. So now getting into the base technology of Twist, I always say the best companies out there really understand the one thing they're really good at. And for Twist, that's our ability to print DNA. So we have this amazing silicon platform shown in the middle of the slide where we can actually print up to 1 million individual oligos of discrete sequences up to 300 base pairs in length. So it's an amazing system for making oligopools. And we can actually make clusters of oligopools on this silicon array. And then we can actually elute off those clusters to actually then make those oligos I was talking about before, where we can basically splice them together to make any gene we wish. And we can also use these oligopools to create highly diverse libraries, which I'll talk about in a second. And so Twist Biopharma, we use those two products of Twist, custom clonal genes, as well as highly diverse DNA libraries to help our partners discover and optimize antibodies. Next slide. I'm often asked, like, what are the advantages? Like, why would I want to work with you, Aaron? Like, what differentiates you from all the other antibody discovery companies out there? And I really cite these three main advantages. One is really the breadth of our capability in the DNA space, that we can create large numbers of libraries. Like we actually have 18 different libraries now, whereas a lot of our competitors, it's really difficult sometimes to build libraries, as you'll see in a second. But because we're experts in DNA synthesis, we have this amazing ability to do it on a monthly basis. Second, as you'll see in a second, we use synthetic sequences as we build our libraries. And so those discrete sequences are actually derived from natural human sequence. So even though we're building synthetic libraries, they're actually very natural in their construction. And then finally, I oftentimes really impressed by the amazing ability of Twist to apply automation to the biotech environment. And because of their product focus on making genes, we've actually enabled and actually implemented many of those learnings in our antibody discovery and optimization process. Next slide. And so just at a high level, in terms of those technologies, we have kind of two base platforms within Twist Biopharma. One is our library of libraries, where we have, like I said, 18 different libraries in phage display that have been developed using our synthetic DNA platform. And then we also have a whole platform for antibody optimization. So we can either help people discover an initial antibody lead against any particular disease target they wish. And then finally, if they if they actually already have a lead, but they wanted to say humanize it or optimize it, we can also apply our library platform to help them in that endeavor as well, using our Tau platform. Next slide. Just to give you a couple of vignettes of how we've used this platform to, in this case, help the pandemic as well as other disease indications. I'm going to give you two examples. The first one is addressing COVID-19. We actually, when the pandemic hit, we actually started using our library of libraries to find antibodies to the SARS-CoV-2 virus. We also undertook a campaign to find antibodies to ACE2, which is the receptor for SARS-CoV-2. And from that, we actually discovered over 200 different antibodies in less than 6 weeks. We actually had multiple candidates come from this. Many of them were actually VHH antibodies, which are, again, very similar to the antibodies that come out of llama's, hence my love of llamas, and really showed the power of our platform to create high affinity, unbiased leads that had about a range of different epitopes that had very potent activity in pseudovirus, as well as live virus, as well as in preclinical animal studies. And we're actually moving many of these leads forward as a therapeutic for COVID-19. Next slide. Just secondly, we've also been very focused on pushing forward our internal pipeline. This is one example of an antibody that we've created against ADORA2A, which is in the immuno-oncology space. This receptor is highly expressed in lung, colorectal, and prostate cancer. And we hope that this antibody, which is really first in class, there's no one's ever been able to find an antibody antagonist of this integral membrane GPCR receptor, that hopefully we can actually combine it with standard of care immuno-oncology drugs like anti-PD-1 or anti-CTLA-4. Next slide. And then finally, I love to cite that Twist is very focused on helping others as well, not just doing our own internal work. We have a number of partners where we're helping them using our library of libraries as well as our Tau platform to help them to discover and optimize antibodies. And we're really proud of a lot of our strategic partnerships with, say, Takeda, Invetx, Neogene, all in different spaces of biotech, but it really shows you the power of our platform to help others in different areas of biotech, because they all require antibodies is their initial starting point for whatever place they're jumping off to in terms of addressing disease. Next slide. So just to conclude on the Twist Biopharma story, we're really here to help biotech and pharma partners as much as we can. We have a lot of different avenues that we can do this in terms of licensing our libraries, generating new antibodies, helping others optimize, collaborating on select leads, as well as partnering on Twist drug candidates that we discover internally. And so we really are pursuing all of these in parallel, and we love to collaborate and help others in the endeavor of discovering antibodies, because at the end of the day, we really want to help discover drugs that help people. And so all these amazing DNA technologies, when focused, as we've created them with the people that we have really enabled this, and we're really proud of our successes so far. Next slide. I'll just end with one last little plug for BioRender. I actually wrote a chapter on molecular mimicry over 2 decades ago. Again, back then there was no tools for making great images for publications, and so I actually had to hand draw the image shown on the left. So it took me a while. It took me actually over several days to draw this image by hand. I'm not a great artist. And then I asked the BioRender team to help me create one that was comparable but much better than my hand-drawn image. And you can really see that we've gone from several decades ago, hand drawing images, to where we are today, where we can use this fantastic software to create something that's very simple, much more simplified, very elegant. And you can really see how having these tools that we have at BioRender really allows us to create these images. And I hope you've appreciated that in my talk today, images were really critical in conveying the message. It wasn't so much the words, but the actual physical images help convey the story. And so I hope folks will continue to use BioRender, because it's really important for me and my team as we write papers, have presentations, do all kinds of different marketing opportunities. We really loved having the tools of BioRender to help convey that message. And I'd be happy to answer any questions.
Great, thank you. Well, thanks for sharing your story, Aaron, Dr. Sato. That was really inspiring. Great to include all the personal elements as well. There's a little bit of a delay on the feed, but it looks like we have some questions coming in now. First question, can you tell us a bit more about the llama antibodies? Like what makes them special?
Yeah, so llamas and other dromedary species, and also sharks, they actually make what's called a heavy chain only antibody. So unlike antibodies which have a heavy chain and a light chain like IgGs, llamas actually make kind of like almost like a half antibody, like they don't have the light chain. And so that allows them to create an antibody binding domain that's much smaller. And what we're finding is that they can actually bind other epitopes and spaces on other targets that might not be available to conventional IgG. And actually what we're seeing even with COVID is we're able, like a lot of, not just our lab, but others, we're able to find a lot of really fantastic high affinity, really potent antibodies that are derived from llamas against COVID-19. And so you can really see that having different structures of antibodies that have different modalities give you access to a much broader space of epitopes on different proteins. And so that was actually really key to finding antibodies to a lot of different targets, not just COVID-19, but other integral membrane proteins as well. So that's why we really love antibodies derived from llamas.
Interesting. Fascinating. That's something that I would have known myself. I have a question on one of the slides, maybe somewhat unrelated to the presentation, but something that was included. You mentioned the growth of the inhibitor market on one slide and just like how big it's gotten. I mean, what's driving the growth of that market specifically?
So I think that was relating to the immuno-oncology market. I mean, what we're finding is immuno-oncology drugs, they are very potent. They do have a really great way of blocking specific receptor ligand interactions that then activate your immune system to then potentially eradicate your cancer. What we're also finding, though, is that only a subset of patients really respond to that therapy. But the cool thing is that it's known that you can actually combine many of these therapies and basically get to a broader range of folks that respond to this therapy. So, 1 and 1 definitely makes more than 2. And so that's why there's this drive continual drive to find more and more inhibitors in this space, because you can combine them together to get to a larger population of people that are suffering from cancer. So I think that was the main driver for why people are still looking into antagonists in this space.
Great. Thank you for that clarification. Our next question, thank you, Nathan, for asking. Can your libraries be used for the development of, I'm going to mispronounce that.
Aptamers.
Yeah, aptamers as well.
So aptamers are basically, the DNA itself is the binding unit. So you could create libraries of DNA molecules in an oligo pool. So we can actually generate highly diverse oligo pools. And so you could use that as an initial starting point to develop an aptamer library. You also could take multiple oligo pools and stitch them together to create a highly diverse DNA library, which you could use again as an initial starting point for an aptamer library.
Great. Thanks for the question, Nathan. I'll let you field these questions, Dr. Sato, because it looks like you can see them as well as I can.
I wonder did you use any computational tools to design it? Yeah, well, of course we did. So we did a lot of work so we get, of course, human repertoire data, and then we use that data and we have to mine through it and really comb through and look at specific germ lines, look at cluster analysis of specific sequences. So we do a lot of informatics work up front, as well as like filter away sequences that might have some issues with developability. And then we use all that data to then inform the design of a synthetic library. So computational methods are really important for us. And I'm even trying to think about ways computationally to discover novel sequences as well as to optimize sequences after you have them. And so we have a lot of efforts on the discovery front as well as the optimization front using computational tools. How popular is antibody therapy at the moment? Again, one of the nice things about COVID-19, and there's not many nice things about it, but one of the nice things I loved about it is actually it made antibody discovery pretty popular. I think I also think it made the idea of being a scientist popular. Like for the first time, like my kids actually thought it was cool to dab as a scientist. So, and the fact that, I also discover antibodies, and it relates back to like vaccines and all these amazing things that are helping to eventually get rid of or lower COVID-19, has been really helpful to, make science cool, which I always thought it was, but it certainly has gotten more and more popular. And of course, antibody therapy should be a popular subject because, it's helping us in so many different ways, not just for infectious disease, but for oncology, inflammation, and even in another area that where Twist is getting into in collaboration with InvetTex is animal health. Can you use antibodies to help dogs and cats? So, we think there's all kinds of different areas to use antibodies, and of course, I think they're amazing and are here to stay. Are llama antibodies worse at recognizing conformational epitopes than IgGs? I'd say they're comparable. Like I said before, I think llama antibodies can sometimes access other certain epitopes that conventional IgGs can't. And it's possible that the reverse is also true, that there are some epitopes that IgGs can access that llama antibodies can't as well. But that's why I think it's good to have both base technologies at your disposal, which we have within Twist Biopharma. Are there any ADME rules when designing antibody drug candidates? ADME rules. So again, I'm a little out of my field, but yeah, so we definitely try to incorporate as much rules into the understand the developability of our antibodies to make sure that they are stable, that when we inject them into people, they stay intact, and also, we have to understand how long they perpetuate in the blood, as well as, do they have any toxicity associated with on-target effects? And so that's all part of the journey of discovering an antibody and developing it. And certainly those are all important along the path to discovery and development. When the libraries are created, constraints to filter the design biologics is essential. How do you do that in Twist Biopharma? The way we do it is basically by using oligo pools to generate libraries. We have a lot more tighter control over how we build the libraries. So unlike in the past where you would typically use a degenerate oligo to create a synthetic library, here I can use pools of individual sequences and then splice them together to create a library. So as an antibody engineer, I love using oligo pools to do these libraries because it allows me to think about a library and then actually create it. Whereas before, there was a disconnect in what I could think and what I could create because degenerate oligos don't get you there all the way sometimes. So that allows me to be much more creative, create libraries much more readily that are higher quality using oligo pools versus the whole kind of older approaches. Great. Thank you, Natalie.
Well, a huge thank you to Dr. Sato, and science is definitely cool again. Congratulations on your continued success at Twist Biopharma.